Our first speaker, Dr. Aritro Sen, if you would please join us. Dr. Sen is an assistant professor with the Division of Medical Endocrinology and Metabolism with the Department of Medicine at Rochester University School of Medicine and Dentistry in Rochester, New York. He's also an assistant visiting scientist with the Center for Human Reproduction right here in New York. Welcome.
Thank you. So I'd like to thank the organizing committee for giving me the opportunity to present our data here. I know it's a clinical forum. I've been given the charge to do the slow transition. So what I'm going to do is I'm going to prepare you for what's coming next. I'm going to show you some data that our lab has done in animal model, looking into androgen actions on ovarian function, and I have nothing to disclose. So whenever we talk about androgens, the thing that comes to our mind is that it's a male hormone. And other than androgens being considered as a precursor for steroidogenesis or estrogen pathway, a few years back, little was known about what androgens were doing directly to follicular development or ovarian physiology. In fact, androgens were considered primarily to be detrimental for women's health and often associated with, hirsutism and PCOS. So we started working in this field about 6-7 years back, and we started off with a very simple question. And that was, if too much androgens are causing dysregulated ovarian function, what happens if you have very less androgens? And to answer this question, we developed a granular cell-specific androgen receptor knockout animals. And this we did about 6 years back, and we published this paper in Molecular Endocrinology. And I'm not going to go in details because this has been out for a long time. But what we found is that if you get rid of androgen receptors in the granular cells, these animals are subfertile, have defective follicular genesis, and develop premature ovarian failure. So what we found is that even though high androgens are detrimental, you need direct androgen actions in the ovary for normal follicular development. And as the debate was going on, that your work needs to be repeated, in the scientific field. So to we are very happy to see that 2 years later on, Christie, Walters, and Handelsman from Australia developed a very similar granular cell-specific knockout model, and they repeated exactly what we were seeing. So what we found in these animals were that these animals were subfertile. They had fewer puffs, and the litter size was significantly lower compared to wild type and heterozygous animal, and their fertility further decreased as they aged. We also found that these animals were, So these animals were not cycling properly, and when we looked at the follicular development, what we found is that these follicles, some of these preantral follicles were stuck in these animals. Oops, it's very confusing. So most of the preantral follicles in the knockout animals were stuck. They were not moving forward. As a result of which, you had fewer antral follicles, fewer CL, but you had significantly high level of atresia. What we did, we took these follicles out and cultured them in vitro for 4 days. And what we found is that these follicles that we isolated from these knockout animals had an inherent difficulty or inherent defect, and they were growing very slowly. Also, these animals ovulated very few oocytes, which tells you why they were subfertile. They also had high level of FSH, which was a very classic sign of premature ovarian failure. So what we got from this knockout animal model is that androgens were important for two places. One, moving these follicles from preantral to antral stage, and also from preventing follicular atresia. Now, this has been out for a long time. The question now that we faced is basically the mechanism. How is it doing? How is androgens promoting follicular development and also preventing follicular atresia? So about 4 years back, we published this paper in PNS, which offered some insight about how androgens were acting in regulating normal follicular development. And I'm not going to hit you with a data bus. What I'm going to show you, because this has already been published, is basically the model of what we have found. But before that, let me give you a little bit idea of how androgens or steroids work. So as all of you know, steroids, the physiological actions of steroid acts by binding to its receptor, which acts as a transcription factor and causes gene expression. This is the classical way how steroids or androgens work. But in the last 10 or 15 years, it's well established that steroids also induces a membrane-initiated signaling pathway. And what we and many others have found across tissues and cell types is that there's a very well-established synergy between this extracellular pathway and the intracellular pathway that causes the physiological actions of steroids. Now, this is the basic steroid biology. What we found is that androgens in the granulocyte cells has the synergistic effect of nuclear and extranuclear pathway and induces this microRNA called microRNA 125B. And this microRNA targets pro-apoptotic proteins that leads to lower follicular atresia. Now, I'm not showing you the data. If anyone wants to see the data, we can talk about later on. For time's sake, I'm just moving on. But what I'm going to show you is two key experiments that we did. One, what we did is there's a survival surgery. In mouse, we injected this inhibitor. This inhibitor of this microRNA in one of the ovaries. So we can take the ovary out, inject it in the ovarian bursa in one ovary. And the second ovary of the same animal, we injected vehicle. And this is an example where we injected India blue ink, blue ink in the bursa. And as you can see, it only goes into the ovary. So it's very specific. It's a survival surgery. And after 4 or 5 days, what we do, we take the ovary out. And we looked at this microRNA, and we were successfully able to downregulate this expression of this microRNA. And you see all these pro-apoptotic proteins are upregulated. So this microRNA, actually what it does, it targets these pro-apoptotic levels, decreases it, helps the follicles to survive. When you block this microRNA, that's what we did, you are seeing these proteins to be upregulated. And what we found is that when we looked at the ovary, the amount of atresia, this is a tunnel assay, the amount of atresia was significantly higher in the ovary that was treated with this inhibitor of the microRNA. But the corresponding ovary, which is on the same mice, had significantly lower amount of atresia. So this was how it's protecting follicular atresia. Now what happens? How does it protect follicular growth? But before I go to that, what we did, once we have found this, these are normal animals. We went back to our knockout animals. And what we found is that indeed in our knockout animals, where we did not have any androgen signaling, this microRNA was lower, the same proteins were upregulated and had significantly higher rate of atresia. So the question now is, how do androgens promote follicular growth? What we found from our knockout animals is that if you block the androgens, the preantral follicles are not moving over to antral stage, right? So the idea is, in normal mouse, what happens if you give androgens? Then more preantral follicles should move over to antral stage, and you should see more antral follicles, right? So we did an ex vivo experiment where we took the mouse ovary out and cultured them in vitro with androgens. And we used DHT, and there was a reason why we used DHT, because if you use testosterone, it gets aromatized to estrogen, and then you won't know whether it's an estrogenic effect or an androgenic effect. So that's why we used DHT, because it's a non-aromatizable androgen. And as you can see, we have the quantitative data, but I want to show you the picture. And as you can see, with DHT treatment, you have much more antral follicles compared to the control, which had a higher number of preantral follicles. Next, what we did, we found that androgens are promoting FSH-induced growth. We took the follicles out, exactly what I showed you previously, and grew them for 4 days. in presence of FSH. Now, FSH alone caused the follicles to move forward. But in presence of DHT, about 25 nanomolar, the advancement was much faster. So what androgen is doing is it's promoting FSH-induced follicular growth. Now, the question is, how is it doing it? And what we found, and this was surprising, is that androgen induces FSH receptor protein level, but it does not have any effect on the mRNA level. So this is a pure translation, non-genomic transcription independent effect. How is it doing? We don't know yet. We are still working on it. But what androgens are doing is basically it's inducing the translation of FSH receptors in the granulocyte cells. So this told us that maybe what androgens are doing is basically it's ultimately causing those follicles to be more sensitive to FSH. And that was the case. We did a antral follicle count in an ex vivo experiment where we took the ovaries out and treated them with different concentration of FSH in presence or absence of DHT. And as you can see, DHT is increasing, the androgens are increasing the sensitivity of the follicles to FSH actions. And how it's doing? By increasing FSH receptors. So, in summary, what I've showed you so far are two mechanisms. One, where androgens, through the increase of this microRNA, are regulating or decreasing follicular atresia. And the second one, through increasing FSH receptor protein levels, are controlling the preantral to antral follicle growth. So at this point, we were stuck. Because, before that, in the last couple of years, there's been a lot of studies showing in the clinical data that's was coming out, and Dr. Glacier will talk about this more later on, that if patients are given pre-treated with androgens, there's more oocyte yield and it helps in the IVF procedure. So we wanted to replicate this in an animal model and see if this was occurring or not. So we did exactly what some of you guys do in the clinic. We treated these animals with DHT for 3 days and then superovulated these animals and collected the oocytes. And as you can see, about 0.25 nanomolar of DHT significantly increased the number of oocytes that was retrieved irrespective of the age. But if you give higher amount of DHT, there was a trend that the number of follicles were decreasing. So from this, we concluded one thing, that maybe balance is the key. If you have too much androgens, it's causing things like PCOS. But if you have too less androgens, it might also cause defects in your follicular development, maybe leading to premature ovarian failure or diminished ovarian reserve. So what we found is that balance with androgens is the key. Now, at this stage, we were stuck because the problem here is that androgens are mainly considered as, androgen receptors are mainly considered as transcription factors and is acting through genes, right? But most people who are working in the field, if you see, in the ovary, there are very few androgen-specific genes. There have been a lot of work that has been done with microarray studies and RNA-seq studies in PCOS patients, in animal models and all. And all the genes that have been found that have been regulated by androgens, none of them are direct targets of androgens. That means none of them have ARE, androgen receptor binding elements on their 5' end. Very few. So what's happening? So we proposed a simple hypothesis that instead of regulating genes that's containing AREs, androgens might be having a global effect on gene expression. And it's a very limiting factor in this field if you're studying the mechanism of androgens and the follicles, because you don't have any downstream genes that are directly regulated by androgens. So we had this idea that it might be globally affecting. Now, gene regulation can be regulated at different levels, and one of it is basically the accessibility of transcription factors and controlling elements to the 5' end. Okay? And accessibility is broadly regulated by chromatin compaction. And one of the major proteins of chromatin compaction is called polycomb binding proteins. And one of them is EZH2. What EZH2 do is, now I'm going to epigenetics, and you have seen a little bit more in the earlier talks. But what EZH2 does is that it causes methylation on histone 3 on the lysine 27 residue. And this is a repressing marker. What it means is that when this marker is present, transcription factors cannot go and bind. So, for example, you are sitting on that chair. If you're sitting on that chair is not free for the transcription factors to come and bind. Only when you move, then only the chair is free for the transcription to come and bind and cause gene expression. Okay, so wherever there's EZH2, wherever this mark of trimethylation on lysine 27, it's a gene repression marker. So you have to get rid of EZH2 in order to get rid of this mark. Open up the chromatin and let the transcription factors bind, causing gene expression. Now, if you look at EZH2, it has got a number of phosphorylation sites. One of the phosphorylation sites, which is S21, has been shown to be phosphorylated by AKT. And there's a science paper a long time back which shows that when EZH2 is phosphorylated at S21, it prevents its enzymatic activity. Okay? And Sharon Walker's lab in Houston has shown that estrogen through the PI3 kinase AKT pathway can phosphorylate EZH2 at S21 and prevents its action. So we wanted to see whether the same thing is happening with androgens in the ovary. And to be short due to time, all I'm showing you is basically the model. And what we found is that androgens, through the androgen receptor and that transactivates the EGF receptor, activates the PI3 kinase AKT pathway and phosphorylate EZH2 at S21. Now this is a very fast and short process. We see this within half an hour, 15 minutes of androgen treatments in granulosa cells. But what we found is that there's also a long-term effect where androgens are regulating EZH2. And what it's doing is that it's inducing through its classical pathway another microRNA called microRNA 101 that targets EZH2 and decreases its level over the long time. Now, so we figured out the mechanism, but the question is, what are the downstream targets of this mechanism that is physiologically important? And in our search, what we found in prostate cancer field is that RUNX1 is an EZH2 induced target. Now we were very excited about RUNX1 because we already knew from Thomas Curry's lab in Kentucky that RUNX1 is very important for ovulation and this granulosa cell to luteal transition. So Thomas Curry's lab has shown this. He has done a beautiful work. So what happens with RUNX1 is that just prior to LH surge, this RUNX1 gets activated in the granulosa cells of preovulatory follicles. Okay, and the LH-induced signaling pathway then induces RUNX1 that is very important for ovulation. So what we did, we repeated exactly the same experiment that Tom has done in his lab. What we did is that we took granulosa cells and cultured them with HCG for 4 hours. And we saw about 4 to 5-fold increase in RUNX1 expression, which was exactly the same what Tom was doing. Now, when we pre-treated these cells with DHT for 48 hours, nothing happened. But when we pre-treated these cells and then treated them with HCG, as you can see, there was a significant rise in RUNX1 expression. Now, we then knocked out EZH2 in these cells. And as you can see over here, just by knocking down, this mimics the effect of DHT. So what I'm showing you here is that the DHT-induced regulation of EZH2 is basically regulating RUNX1 expression. So we wanted to further establish that the mechanism that we are seeing, like the phosphorylation of S21 and the microRNA, are actually involved in RUNX2 expression. And what we found is that we know that androgens are regulating EZH2 through PI3 kinase and microRNA. Now, is it important for RUNX1 expression? So you have already seen this, okay, in the previous slide. What we did is that we mutated this S21 to alanine so that it cannot be phosphorylated anymore. And then we took this mutated form and overexpressed them in the EZH2 knockdown cells. So we have already knocked down the endogenous EZH2 and overexpressed this mutant in these cells so that the S21 cannot be phosphorylated. And as you can see over here, when we mutate the S21, you lose the positive effect of DHT priming on RUNX1 expression. We did a very similar experiment with microRNA 101. We knocked it down over here. And as you can see, when we got rid of the microRNA 101, this also makes the DHT lose its effect on RUNX1 expression. So, finally, we went and looked at the promoter region of RUNX1 over time with DHT treatment. And DHT causes hypomethylation of RUNX1 promoter. So what it's doing is it's inhibiting the EZH2, getting rid of H3K27, causing hypomethylation that's allowing factors to come and bind on the RUNX promoter and causing the gene's expression. So this is basically, it's DHT is causing hypomethylation of RUNX1 promoter region over time. So in summary, what we have found is that androgens, through a fast and short mechanism, phosphorylate EZH2 as S21 that prevents its enzymatic activity. In addition, androgens, through inducing microRNA 101, targets EZH2 that opens up its promoter region, causing LH-induced transcription factors to come and bind to it, causing RUNX1's expression. So we have done this, but what is the physiology of it? We have seen this, we have done the RUNX1 and all, but you have to understand that we have to know what is the downstream mechanism, what is the physiology of it. So we did a very similar experiment that I showed you previously. What we did, we injected microRNA 101 inhibitors in the bursa, let the mice survive for 3 or 4 days, and then we superovulated these mice and collected the oocytes. And what we found is that when you block this microRNA, the number of oocytes that's ovulating are significantly lower. In addition, when we looked at the expression of microRNA and RUNX, this was significantly lower. But what we found is that there's another gene, PTGS2, which is basically COX2, which is known to be induced by RUNX1 because RUNX1 is a transcription factor. And COX2 is very important for ovulation. That's also getting downregulated. Now, once we have done this, we went back to our knockout animal model. And what we found is that indeed in this knockout animal models, when we superovulate these knockout animals, there was significantly lower level of oocyte ovulation. MicroRNA 101, RUNX1, and COX2 were also significantly downregulated. So in summary, what I'm showing you is that what androgens are doing over here is that it's not directly regulating ovulation, but what it's doing, it's preparing epigenetically as it's moving, the follicles are moving, it's preparing the promoter region of one of the, so I'm using RUNX1 as just one example, okay, downstream example. We are in the process of doing CHEAP-6 studies and all, which will give us a global idea of what androgens are doing, but here I'm using RUNX1 as a downstream target, and what I'm showing you is that while androgens are not directly regulating RUNX1 expression, it's preparing its promoter region, removing this epigenetic marker, causing LH-induced transcription factors to come and bind, and inducing RUNX1 and COX2, which is basically important for ovulation. And with that, I'd like to thank the people who have done this work in my lab, primarily Emily Hayes. She's an excellent undergrad who has worked in my lab for 4 years now. Shouting Ma, Annie Biswas, and Hen Present, my collaborators at University of Rochester, and I work very closely with Dr. Glacier, Albertini, Dr. Kushnir, and Dr. Barat at CHR, and the funding agencies. Thank you.
Thank you very much. We have 2 or 3 minutes for questions from the audience. Any?
I have a question.
Please.
Yeah.
So introducing this androgen effect at that transition from a pre-antral to antral stage, and given what you've shown about the impact on proapoptotic genes and preventing atresia, how does this fit into the classical Aaron Schway and Hakan Billig model that ascribed most of the anti-atretic activity to the aromatase activity and the production of estrogen?
So it's all balance is the key. If you have too much androgens, that's what's happening. But what we are saying is that you need androgens, but everything is in moderation. You need very less amount of androgens to work a normal thing. So what I'm showing you is nothing wrong. What I'm showing you is basically if you have a normal level, very low levels of androgens, it's normal. The moment you have more androgens, then what's happening is causing these follicles to move forward, exactly what androgens are doing, but you have more androgens, so it's moving more forward, and then it's getting stuck somehow, okay? And that's why it's dying off. So everything is in balance. Too much, too less.
So it's secondary to the local effects of estrogen at that point.
Exactly.
Basically. Okay.
And one more question, I believe, right there. Thank you.
Thank you for your presentation. For how long do you have to give androgens to have a real good effect on the ovary?
So in the mouse, what we have seen is if we give DHT injections for 3 days, 1 injection for 3 days, and then we superovulate, then we can get a higher amount of oocytes. Now, I'm using DHT, which is much more potent than DHEA or testosterone. But I think Dr. Gletscher will show some of our data from the mouse where we have also done DHEA stuff. But it's all about how potent it is. That's why. So we have given DHT 3 days. It does pretty much. We see a difference in the number of oocytes being retrieved.
Very good. Well, thank you
you
very much, Dr. Sen, and thank all of you for participating.